So this video will wrap up Chapter 3 on microscopy and staining by talking about some of the techniques that we'll be using in this course and that are used by microbiologists in order to learn more, to define what kind of organism they're dealing with. So the first technique, and you have some experience with this already, is in making a wet mount. Wet mounts are allow us to look at live specimens, and so with making a wet mount, you put a drop of the fluid with whatever the microorganism is onto a slide, and then being very careful to slowly lower a cover slip onto the slide. Then you seal the drop of fluid between the slide and a glass or plastic cover slip. And so this lets you look at, it forces the specimens into a relatively thin film of water that's found there between the slide and the cover slip, so they are relatively easy to maintain and focus as you look at them. And sometimes some of the protozoa, especially, that we might be looking at using a wet mount technique, the protozoa can be moving very quickly, and especially if they're at high magnification, it's hard to keep up with them. And so you can use a viscous solution of a material called methyl cellulose. It goes by the trade name of proto-slow because it slows down protozoans, and it makes the fluid in under the wet mount a lot stickier and a lot harder for the animals or the organisms to swim through. So that's a wet mount. If you really want to look at the swimming behavior of the organisms, then you can use what's called a hanging drop technique. And this requires a special kind of slide called a depression slide. If you look at it in side view, it would look like this. I've got it shown here. And so in order to do the hanging drop, you put a drop of whatever the fluid is with the specimen in it. You put that onto a cover slip, and then put 4 little dabs of Vaseline at the 4 corners of where the cover slip will be, and then invert or turn over the depression slide and smush it against the cover slip so that that little drop is there at the depression. And then you can flip the whole apparatus over now so that the cover slip is back on top again, but the drop of fluid will remain suspended beneath the cover slip. And then you can add immersion oil if you need it, depending on how much you're going to try to magnify the specimen as you look at it, and then can look at it underneath the scope. And that allows you to observe motility in flagellated specimens of bacteria or ciliated or flagellated protozoans. A third way to get a specimen to stick onto a slide is to make a smear, and this allows you to look then at dried or preserved or dead specimens. So using a tool called a loop, it's just a handle with a thin wire coming off of it and a loop at the end of the wire, you can grab a sample of bacteria that are found either in a liquid medium, in which case you just take a couple of loopfuls of the specimen and smear it out onto the slide like we're seeing here. Or if it's something that's grown on agar, then you grab a little sample of the specimen and then smear it into a drop of some distilled water or clean, purified water, so you're not adding new specimens to the sample. So you can do this smear technique with either liquid, the bacteria that are grown in broth, or black bacteria that are grown on agar. You allow this now very thinly smeared and spread out specimen to air dry, and then very briefly hold it over a flame. So what I usually do is pass it through the flame 3 or 4 times, and that not only kills the microorganism because you've heated it up pretty hot, even though you're just passing it through briefly, it still gets the slide very hot, and it causes the capsule of the bacteria to adhere very strongly to the slide. So you can rinse the slide if you need to as part of a staining procedure, and the bacteria will remain adhered to the slide. So sometimes that heat fixation will also assist in making the bacteria more likely to take on staining materials. But it can destroy the capsule, so you may see some loss of what the organism looked like in real life. So then you can apply the stain, and you can apply a big puddle of stain to it, and then rinse off the excess and dry the slide, either letting it air dry or squeezing it between the pages of a little pad of very thick paper. It's called bibulose paper. And I'll introduce this to you when you are making your first slide, but it's called bibulous paper, and that blots dry the smeared slide, and then you can you can just pop that dude right underneath the microscope. No cover slip is even required, or you can add additional staining or use other microscopic techniques with this smeared and dried and stained material. So that's a smear. Okay, so we also use stains, as you've seen, or as we've now discussed a little bit, and stains are just dye molecules that bind to and color particular structures in the cell. And the goal of staining is to increase contrast between either the specimen or structure, depending on what you're staining, and the background coloration. So it increases contrast and makes it easier to see details like edges and internal details. And we categorize different stains, or different bacteria, we can identify and classify bacteria based on how they take up different stains. So we talk about that as their staining properties. So there are a couple of different types of stains that are used. A basic stain, also called a cationic stain, because basic materials are also cations. So it has a positive charge. That's what cation means. So a cation has a positive charge. So these basic stains have positive charges, and so they get attracted to structures in the bacteria or whatever you're staining that have a negative charge. So cell membranes, for example, have negative charges, and so basic stains are attracted to and bind to the membranes. So some of the examples of basic stains include methylene blue, crystal violet. Methylene blue stains everything a deep dark blue color. Crystal violet stains things kind of a rosy pink color. Crystal violet, a dark purple color. Saffronin stains things like a kind of a rosy pink, and malachite green, which, as you might guess, stains things kind of greenish coloration, blue-green color. Okay, so there's basic stains. Acidic stains, they have a negative charge, and so they are attracted to any structures in the cell that might have a positive charge. And so some of the acidic stains that we use are eosin and picric acid. Okay, so we can describe stains based on their physical properties, and then we can describe processes of staining. So a positive stain, for instance, would color the specimen so that it contrasts against the background. A negative stain, rather than coloring the specimen, actually colors the background while the organism resists picking up the negative stain. And so this allows you to see especially superficial structures of it, so the shape of the cell, the size of it, or to visualize a little more easily the capsule that surrounds the bacteria. That's a really rigid polymer of material that is produced by the bacterial cell. The capsule is. A simple stain, when we talk about a protocol of staining that only requires one stain, then is referred to as a simple stain. It just uses a single stain. So methylene blue, like you did with your cheek cells, crystal violet, saffronin, they can all be used as simple stains. Differential stains are staining protocol that use two or more stain materials so that you turn some of the bacteria one color and some of the bacteria another color, or turn specific structures within the bacterial cell a different color than the rest of the bacterial cell becomes. So it's used then to distinguish among different organisms or different parts of an organism. And some of the more common stains are the Gram stain, named for Hans Christian Gram, and the Zeil-Nielsen's Acidfast stain. So those two are widely used to help characterize or identify different organisms from each other. So here we see an unstained material. So these are the unstained cells here, and this is another set of cells there. And here we see those larger cells after staining. And here are the smaller cells with now darker staining material in them. So this is a simple stain that then highlights different structures of the cell. And here we see an example of what you see following Gram staining. So these are two different rod-shaped bacteria that we're seeing in this sample. Some of the rods are a dark blue, or actually, if you could see this in real life, it would be a deep purple. And some of them are a lighter rosy pink. So we've got some Gram positive. We have Gram positive rods here. Those are the dark purple ones, and Gram negative rods are the lighter rosy colored rods there. So differential stains require two or more stains so that you can produce a difference between different groups of bacteria or different structures in a bacteria, while a simple stain just uses a single stain in the protocol. So how do you do Gram staining? Well, you make a smear like we talked before, and you heat fix the smear, and then you apply a very deep dark purple stain called crystal violet, and that stays on the smear for about a minute. And then you pour off the crystal violet, but you don't rinse yet. Pour off the crystal violet and follow it up with a minute of iodine. So we drop some iodine then, which is kind of a yellowish colored pigment, but the job of the iodine is not necessarily to add color here, but it will bind to crystal violet that's made its way into a capsule that is really thick. So it binds to that crystal violet and makes that molecule even larger. And then we decolorize the specimen with alcohol, and that rinses away crystal violet from any structures that don't have the mordant effect that iodine causes. And so for structures that have a relatively thin capsule, then the crystal violet washes out, but structures that have a thick capsule around the bacterium, they are not decolorized very easily. So then you rinse everything with distilled water, and that takes a little bit to get everything rinsed off. And that alcohol, boy, it washes a lot of the crystal violet away, but you can see that there's still a little crystal violet left, kind of coloration here on the slide. And so you rinse that with distilled water, and then you use what is referred to as a counter stain. And in the case of the Gram stain, saffronin is the counter stain. Saffronin makes everything sort of a light pinky color. And so you apply saffronin for about 30 seconds, and then give it a little water rinse. That doesn't decolorize, it just rinses the excess safranin away, and then blot it dry with some bibulose paper, and then you look at it underneath the microscope. And anything that is dark purple is gram positive, because the mordant has helped the primary stain of crystal violet to be caught up in the capsule. And anything that is pink stained here is gram negative, because it rinsed away the crystal violet and we only see the safranin color. Now safranin has stained the gram positive little cocci, the little balls there. It stained them, but because the crystal violet is so dark, you can't see it. So everything gets stained by the safranin, but only the capsules that have also taken up the mordant of iodine. And again, a mordant is just an additional dye that helps the original dye stick to and in some structures. So you can only see the safranin in the decolorized cells, because it's a light purple that's kind of covered over by the dark, I'm sorry, it's a light pink that's kind of covered over by the dark purple of the crystal violet. Okay, so that's how the gram stain works. And it sorts bacteria into 4 groups. Gram positive, they're the ones that are deep violet. Gram negative, they're the ones that are kind of pinkish. Things that are gram non-reactive, and so they don't pick up any color at all. And then sometimes a group can be gram variable, meaning that some of them are violet and some of them are pink. When we get this result of gram variability, the first thing that we suspect, though, is that it's an old culture, because in gram staining, if you have an old culture, one that is more than about 24 hours to 48 hours old, then they often become gram variable as more of these older bacteria start to be hanging around in the solution. Okay, so that's the gram stain. The Acidfast stain protocol, developed by Zeal and Nielsen, it's used to distinguish between bacteria that have a specific material in their capsule as well. So it can tell whether or not a material called, I'm looking for the name of it because I can't think of it all of a sudden, and where is it? It's in the next slide. I'm going to look ahead for just a second. It's mycolic acid. It will react differently if the capsule has mycolic acid in it or if it doesn't have this mycolic acid in it. So just like with the gram stain, you start by making a smear and heat fixing it, and then you start with a primary stain of carbol fuchsin. So that's a kind of a bright, annoyingly bright pink color, carbol fuchsin, and then you heat it very gently while the primary stain is being applied. Then you give it a rinse with water and use, this time, hydrochloric acid, a 3% solution of hydrochloric acid, to decolorize the material. And so you decolorize with hydrochloric acid, and then you counterstain this time with methylene blue. And if there's mycolic acid in the capsule, then the methylene blue will not stick to it. So so we use this term acid fast, and it's in the same idea as being color fast. So if something is color fast, it doesn't rinse away the dye that's in it. So an acid fast bacterium is one that doesn't rinse away this carbol fuchsin even when you decolorize with acid. So the color stays. So in the case of the experiment that we have here, we've got some rods and some cocci, the little balls. You apply heat to it. Heat acts as the mordant in this case, really making the carbol fuchsin stick in the cell membrane of those with mycolic acid. Then you decolorize. Notice now we can't see. Now we can't see the little cocci, the little balls. And then follow that up with a counter stain of methylene blue, and anything that is acid fast will remain hot pink, while anything that is not acid fast will now be that dark purple color. So it sorts bacteria into two groups: the acid fast ones with mycolic acid that are kind of a hot pink red color, and the non-acid fast ones that have the deep purpley blue of the crystal violet. And because they don't have mycolic acid, then in their cell membranes, the hydrochloric acid will wash the carbofusion from the membrane. I've been saying the capsule, but it's in the cell membrane. I apologize for that. So this allows you to distinguish between or those species of mycobacterium that are the causes of both tuberculosis and leprosy. And so here are some images of the purpley color of carbofusion in some acid-fast bacilli, the rod-shaped bacteria. Okay, and then we wrap up with a little bit about talking about how we can stain particular cell structures. So there are flagellar stains. In bacteria, flagella are very, very thin, hard to see structures, and so coating them with a dye or a metal, something called a flagellar stain, helps to make them much more obvious. And so you can see here the flagella of these rod-shaped bacteria that have picked up this flagellar stain. There are some bacteria as well that produce something called an endospore, and there's a specialized staining technique that will highlight the endospore. It's called the technique is called the Schaefer-Fulton spore stain. By the way, we should probably explain what an endospore is. It's a chunk of bacterial DNA that's dormant and then surrounded by layers and layers of really resistant materials. And we've talked about them a little bit in Chapter 1, so you've already been introduced to the idea of the endospore. And so the vegetative cells, the growing cells, will form them as the conditions in the bacteria's environment become adverse. And they're very resistant to heat and to time and to all sorts of adverse things, and will germinate later when given the right conditions. And so endospores tend to resist staining by most techniques in the same way that they can resist time and heat and all that. And so the protocol requires submerging the cells in a solution of malachite green and then heating it up so that the solution is steaming, and allow it to set for 10 minutes. And this gives time and heat that allows the stain to penetrate the endospore wall. So first you use the primary stain of malachite green to turn the endospores green, and then counterstain with safranin. And so what you see is a bright green, that malachite blue-green color endospore in a safranin red cell. So we can stain particular structures as well to get a better idea of what they look like. So there you have it, a bunch of the techniques, and we'll be practicing these techniques in the next couple of weeks. So you'll get a chance to play around with some of these, especially that gram staining technique. All right, so that does it for Chapter 3, and you should now be ready, hopefully, to take an exam on this. So if you're not ready, if you have questions, don't hesitate to contact me. You know the ways to get me: by mail, by phone call, email, phone call, or meet me in office hours.